So this lecture is going to start us off with our section on our water, weak acids, buffers, and the Henderson-Hasselbalch equation, right? So this is going to be the first section, or really the first portions of this lecture are going to be additionally review from Gen Chem 2 and maybe some of your other prerequisite courses about weak acids, incomplete dissociations, and some of the different types of bonding, right? And how those are going to impact biochemistry as we move further into our topics in class. So just a general overview, right? We're going to discuss a little bit about biochemistry and water. As biochemists, why do we even care about water? Some comments about acids, bases, and buffers in biochemistry. And then in this lecture slide, we're going to touch on some basic aspects that contribute to how we use and understand the Henderson-Hasselbalch equation. But ultimately, we won't get to using that in this lecture, in this specific lecture. So we'll cover that in our next sequence. So recall that bonds or intramolecular structure, the components that contribute to the intermolecular structure of molecules and electrolytes, ultimately determine how specific molecules and ions interact with other molecules and ions, right? So remember the intramolecular structure, bonds, electronegativity, those kinds of concepts. Remember those lead to how molecules, again, electrolytes, interact with other molecules. So intramolecular structure leads to intermolecular forces and organization. So, generic review, right? Recall ionic bonds. In ionic bonds, electrons are donated or stolen between a cation ion, such as sodium, and an anion, such as chloride. And as that occurs, we get a positive ion, negative ion that will be attracted through electrostatic interactions and form crystal lattice structures. Covalent bonds, right? Recall from our previous lecture, covalent bonds are when atoms share molecules in between each other and form covalent bonds, whether that's a single bond, double bond, or triple bond. And then depending on how those electrons are shared, that can lead to permanent dipole moments, temporary dipole moments, again, some of the concepts from our previous class, and that can lead to regions of a specific molecule that are electron rich or partially negative, and then electron poor or deficient that are partially positive. And since we're talking about water today, in molecules where this disparity in electrons is due to a large difference in electronegativity, remember with oxygen, nitrogen, and fluorine, that can lead to a special type of intermolecular force known as hydrogen bonding, where those hydrogens are left, again, more positive than other types of dipole-dipole moments, and that allows them to interact more strongly with other partially negative regions of other molecules. In this case, a strongly partially positive hydrogen is interacting with a strongly partially negative oxygen from two different water molecules. Right? And so we focus so much on water in this class because water is fairly unique in its electrochemical structure, and that leads it to having unique, and for the way life developed on Earth as we know it, useful things for life on Earth, right? So water's electron geometry is obviously tetrahedral, right? We have oxygen at the core, we have our two hydrogens that come off bent, and then we have two lone pair electrons up above our hydrogen atoms here. But its overall molecular geometry is bent, right? And bent out of the normal 109.5 degree angle that you would expect to see in something that has tetrahedral geometry. Again, remember that's because those electron pairs up there actually have some extra repulsive forces and are actually pushing these hydrogens closer together than you would expect them to be at 109.5. Right? So remember, also water is highly polar, so if placed in an electrolytical field or an electrical field, the positive ends, so the hydrogens, would align with the negative portion of that electrical field, right? In this case, this would be the cathode, anode. Remember, cation ions travel towards the cathode, and ions travel towards the anode. So water would align that way, right? And again, we just covered the basics about hydrogen bonding between water molecules. And just to give you some data to back that up, down here we have heat of fusion values in calories per gram and joules per gram. So just to drive home, the differences in hydrogen bonding versus other dipole moments between molecules and intermolecular forces. Water has a very strong heat of fusion here at 80 calories per gram, 335 joules per gram, versus if we go down the periodic table, right, sulfide and selenide, who have much lower. So this means that the strength of the intermolecular forces between water, right, this positive hydrogen interacting with this negative oxygen, are much stronger compared to the way a sulfur molecule would, if we replace this oxygen here with the sulfur molecule, how that would interact with a hydrogen, even though there would still be a permanent dipole moment there for sulfur. And so comparing over here, right, just I want to emphasize, again, based on or following up on some of our previous content from 2 lectures ago, hydrogen bonds are weak compared to covalent bonds, right? It takes an enormous amount of energy to break a single covalent bond. Hydrogen bonds are still mostly electrostatic, but they are very influential on intermolecular organization and the strength of intermolecular forces between molecules. So over here, we have heat of vaporization. So this is actually how much energy it requires to take, how much energy in joules it requires to vaporize water, right? So in this case, 1 gram of water. So this is how much energy it takes to convert liquid water into gaseous water, right? Heat of vaporization. So up here we have water with 2,260 joules per gram, right? Quite a bit. Whereas if we keep going down, methanol, down here in the bottom left, which also has hydrogen bonding, but it doesn't have, right, instead of having a hydrogen over here, it actually has a methyl group, so it's not quite, the distribution of electrostatic charges or dipole moments across this molecule aren't quite as strong or organized as water. Ethanol, that trend continues. Propanol, that trend continues, right? So we see a decrease in the amount of energy it actually takes to vaporize those, essentially saying these guys are going to boil at lower temperatures, again, or convert from liquid to gas at lower temperatures than water. All right, methanol, ethanol, propanol, butanol. And you can see again, as we make that hydrocarbon tail longer and longer, the strength of the intermolecular forces or the strength of those hydrogen bonds actually decreases. Now, if we compare butanol to butane, right, so butanol over here on the right versus butane that has no oxygen, hydrogen, or alcohol group on the end, it actually can't undergo hydrogen bonding. So we see a large drop off from 590 to 381. Our boiling point falls very quickly or very dramatically from 117 down to negative 0.5. And then our melting point increases from negative 90 to, it decreases from negative 90 to negative 135, right? So substituting that alcohol group for hydrogen from butanol to butane actually makes a large, drastically changes, again, the intermolecular forces or the strength of the intermolecular forces between molecules of butane versus molecules of butanol, right? So hopefully that really drives home the strength of hydrogen bonds. And again, when we get further into class, we'll discuss these things too, right? In between functional groups where hydrogen bonding plays a large role in how ligands interact with active sites of enzymes, or, enzymes interact with other enzymes. Hydrogen bonding is going to play a large role in many of the interactions we'll see in class. Right? So to reemphasize that, hydrogen bonds and macromolecules. So hopefully everyone recalls that when a solid salt or solid ionic substance, right, such as sodium chloride here, when that's tossed into water, those water molecules actually can intervene and help break up that crystal lattice structure, right? Some of this has to do with entropic forces that we'll cover once we get to our thermodynamics section. And then some other components just happen to do with an oversimplification of like dissolves, right? Right? So we know that salts are often polar. We know water is a polar solvent. Right, like dissolves in like, recall that. And I guess this is technically following up on some of the comments from the previous slide. So water can impact how substrates interact with enzymes, right? So water is going to orient itself and interact with an enzyme based on the specific amino acids that are found in that enzyme. and the specific amino acids on this region of this enzyme, right? And depending on how that all works out, again, every enzyme is going to be a little different. Our substrate can interact differently with that enzyme. And then over here, right, so how alcohol interacts with water, how certain ketones interact with water, and this is what I was referring to earlier. Once we get to actually talking about amino acids and peptide sequences, that hydrogen bonding and what hydrogen bonding is or isn't available can affect how peptides interact with each other, right? Whether that's influencing secondary structures or how peptides can interact with other peptides. And then a stereotypical example that's used to emphasize why certain nucleotide bases pair with others, right? Recall there are hydrogen bonds between nucleotides, right? Adenine, thymine, and then a different set of hydrogen bonds between cytosine and guanine. All right, so salvation spheres here on the top right. Right, so some of you may remember from general chemistry that depending on the charge of an ion, right, again, a cation or an anion, the way water organizes itself around this or orders itself around these ions will differ, right? So recall that the oxygen on water is partially negative, so if placed in solution, a sodium ion that's positive will reorganize and reorient the water molecules around it so that oxygen is closer, right? So oxygen is negative, it's going to be attracted to a positive sodium. That will leave the hydrogen atoms within a water molecule facing outward from a sodium atom, right? Leading to more water molecules orienting in this oxygen to hydrogen, right? And then the number of water molecules that can associate with an ion also differs depending on the size of that ion, right? And the opposite is true for chloride. Chloride is negative, so the positive portion of water molecules, hydrogen, are going to orient themselves towards that chloride ion. All right, and we care about salvation spheres from a biochemical perspective because they can have large influences on what these ions can or can't do, right? Some channel proteins have been built in such a way that depending on the salvation spheres around certain ions can or cannot pass through those channel membranes, right? So we care about the order of water molecules around ions as well as the size of salvation spheres around certain ions, right? And so I wanted to take this opportunity to also discuss one of the reservations we have when we talk about certain components of the inside of a cell. So we discussed macromolecular crowding in one of our previous lectures. So here's our, this image is just to recall that. And then down here, we actually have an example of how water molecules are, believed to behave within a cell, right? So we discussed how small things aren't really free to diffuse rapidly throughout a cell because of things like macromolecular crowding. Over here, this diagram is meant to basically demonstrate that, again, water will orient and order itself around things with charges in a cell. And we'll talk about it later as well, but this allows water molecules to do something we refer to as shimmering, right? As protons can move from one water molecule to the next, that doesn't actually happen because this water molecule or this proton is necessarily diffusing over long distances in a cell, like you may expect if they were free to do so. These water molecules can actually rotate and, more or less allow protons to hop from one molecule to one molecule. So this proton can hop on here. Now this water molecule has become a hydronium molecule with 3 hydrogens, and that hydrogen, the third hydrogen, is actually free to hop to the next water molecule, and so on and so forth. And that's one way that cells have water molecules that shimmer and let things move throughout them very quickly. And this also causes the inside of a cell to behave more like a gel than a true liquid or a true fluid, right? So the inside of a cell is often colloidal in nature rather than liquid. So this slide is just meant to remind you guys about some of the concepts that we talked about from our review lectures, right? Remember that hydrophilic solvents like to dissolve polar substances, right? Hydrophilic, polar, whereas hydrophobic solvents like to dissolve nonpolar substances. Amphipathic molecules are molecules that have both a polar region and a nonpolar region, such as this phospholipid here. Right? It has a polar phosphate group head and a nonpolar hydrocarbon tail. And when large numbers of these molecules are placed together, depending on the solution they're placed in, this can lead to the formation of clusters of lipids, where the hydrophobic portions of those phospholipid molecules are going to orient themselves towards each other, right? Those hydrophobic interactions, because they don't actually want to interact with this water, so they'll cluster together. And then again, depending on what else is going on in solution, this can lead to the formation of micelles, and it actually, with the right level of perturbation, could lead to the formation of phospholipid bilayers, right? And recall from our previous lectures comments about gas transport, right? Some gases dissolve more readily in solutions like water, and then some gas molecules have to undergo that temporary dipole interaction to aid in their, I guess you could say, dissolving into solution.